It says meeting is now streaming live on Facebook.
Wonderful.
Done. Okay.
There we are. We're now on Facebook. Good evening, everyone, and welcome to our July meeting of the Western Pennsylvania Mushroom Club.
Okay, Stephen, we can't see your slides. At least I can't see your slides.
Oh, that's great. That's why.
Yeah, there they are.
Yeah, so I'm Stephen Buckland, the current president, and I've got some updates on our club's activities over the past month. Everyone should have received their July and August newsletter. There's a lot of good stuff in there, including articles on all of our upcoming meetings, walks, and events, including Die Day and the Linkoff Foray. There's also advice for beginner foragers, a review of this year's City Nature Challenge, and lots of other good stuff. Just a reminder, if you would like to submit something to the newsletter, article submissions are always welcome, and you can contact the editor, Cecily Franklin, if there's something you're interested in contributing. Between our last meeting and today, there's been, I think, 6 different walks held in person. First was our annual Dorothy Fornoff walk at Deer Lakes Park. That was led by Richard Jacob. There's a really detailed summary and species list posted to our website if you're interested in seeing what folks found. But this one I thought was pretty interesting. Not every day you see a white chanterelle. So pretty cool. The next day, there was a walk at McConnell's Mill State Park, and that was led by Jerry Sapp Jr. I think there were some photos posted to our Facebook group from that walk as well. So a lot of different Amanita species were found on that one. I was really impressed by all the different Amanitas. Then on June 26th, the Plishkes led a number of mushroom-related events at Blue Knob State Park. So I think there was a walk, a cooking session, and also a cultivation session. So a lot of stuff that they did on June 26th at Blue Knob State Park. Wish I could have made it out to that one. This wasn't a walk, but our former president, Richard Jacob, gave a presentation to the Mycological Association of Washington, D.C. And you can find a recording of that on their YouTube channel. But if you were a member last year, you might have seen a very similar presentation that Richard Jacob gave. But yeah, the Mycological Association of Washington, D.C. has a lot of great recordings up on their YouTube channel. The same night that Richard gave his talk, he was followed by Alan Rockefeller, who gave a talk on the mushrooms of Mexico. So there's a lot of good stuff on their YouTube channel if you are looking for other presentations to watch. On July 10th, there was a walk at Gertie's Woods. Yeah, looks like a lot of people were there. A lot of good stuff was found. Julie, you can chime in if you want. If not, I'll just keep going.
That was me.
Mm-hmm. Oh, that was Kara.
Yeah, that was all me. It was a lot of fun. Highlights, we found a really interesting Pluteus, and it was a Pluteus pipidosus, I believe it's called. Pipidosus. It's been a while. I'm not looking it up right now. but that was a really good one. Other than that, bunch of some of the more common items, slime molds, bunch of, it's pretty dry conditions, but it was still pretty nice.
Sweet. This was Julie's walk, I think, July 16th at Bethel Green. All right, I've got it straightened out now. So yeah. Anything?
It's weird because Gertie's Woods is ALT property, but that one wasn't with ALT. That was with one of the local groups in Millvale. So totally understandable there. But yeah, it was an awesome day. We found over 30 species. I just uploaded them to the website today and sent Richard some pictures. But yeah, probably 34, 36-ish different species in that 2 hours. So it was pretty awesome. I think my favorite we found was the red tinged lepiota, or the ruby lepiota, which was pretty cool. I'd never seen that one before, so I was pretty jazzed. But yeah, had awesome group, good time, weather held out, so can't complain.
You got a jellied colibia that day too, didn't you?
Yeah, we got a colibia on a oak loving
colibia jelly, sorry.
Yep, colibia jelly / colibia cloud / all its other names. But yeah, we found that one on a oak loving gymnopus that day, so we got that one there too.
I'm very jealous.
It was just a couple little spots. It wasn't like anything like too crazy.
Nice. All right, and last but not least, a few days ago, big walk at Heartwood Acres, and that was led by one of our new identifiers, Barbara. And yeah, did you have anything you wanted to share?
Yeah, it was my first walk, so I was a little nervous, but a lot of people showed up and I had lots of help. And yeah, you can see the group picture. It was just like tons of people showed up. And we were in the woods. We had to enter the park from the other side, from the amphitheater side, because that whole mansion road is closed. So I was worried about not finding any chanterelles because they usually grow towards the mansion side, but we found lots of them on the other side as well. So that was good. Lots of species. Richard and I are actually working on that species list right now. I think like I entered mine, he entered his, so he said he was going to merge that. You can just see it on that like bed sheet that I brought. It's just like tons of species. I don't know how many it's going to be, but it was a lot. So lots of chanterelles, lots of boletes. I think my favorite was, and I didn't find this one, but it was the frost bolete. So I was really jealous of that find, whoever found that one, because, and I was joking. I was like, no, you have to give me this mushroom and I have to go back and I have to stage it and photograph it because I don't have a good picture of it. And towards the end, the woman was like, I was like, where did it go? She was like, wait, you were serious? I was like, yes, absolutely. But we didn't do that. We didn't stage it, but that was a great find for me. Lots of the usual stuff like violet-toothed polypores. We found some crown-tip corals, Artemisia spixodatus, lots of russulas, lots of amanitas also.
And we found Enoki coquamenonensis.
Yes, we did. It's a pretty actually common mushroom in Heartwood Acres. I've found it there multiple times. So I don't know if that's pretty rare to find outside, but yeah, that was a good find as well.
Very cool.
Yeah.
Thanks for sharing.
Mm-hmm.
Yeah, really wish I could have made it out to that one. Seems like it was a good time. But yeah, so that was all of our walks over the past month. And here are some upcoming walks with Allegheny Land Trust. There's one at the end of the month at Sycamore Island. That one is sold out. But August 20th at Churchill Valley Greenway and September 10th at Wingfield Pines were somewhat recently listed, so there's probably still space to register for those. And you can register at AlleghenyLandTrust.org/events. And those are going to be led by Julie Travellini, who is our secretary and an identifier for the club. And I think on the 31st, Lamont is going to be helping out identifying. I'm not sure if you roped any other identifiers in for those walks, Julie, but feel free to add any details I missed in the chat. Other upcoming events are our monthly meetings. So this is our last virtual meeting tonight. Beginning next month, we'll be meeting in person, if all goes well between now and then. So yeah, I also wanted to note that log drilling will begin at 6 p.m. So our usual meeting time is 7, but yeah, so in person at Beechwood Farms, and log drilling will start at 6 p.m. for our cultivation meeting next month. Other events that are upcoming next month also is Dye Day. Registration is closed. We got, I think, almost 35 people registered, so really great turnout. A lot of people interested in learning about dyeing. If you missed registration for the afternoon, I think you can still join us for the morning walk, and maybe Judy will share at least some info on like what species you might be able to use for dyeing.
Yeah, the walk is free and it's not limited.
Yeah.
Okay.
So you can still join us for the walk in North Park on Saturday, August 21st, with expert identifier and mushroom dyer Judy McEnroth. So yeah, more details on that are on our website. And of course, we have our annual Gary Linkoff Memorial Foray. The pre-foray walk is on Friday, September 17th at Cook Forest. The foray itself is going to be on Saturday, September 18th at the Rose Barn in North Park. And there will be Science Sunday the following day, also at the Rose Barn. So yeah, registration is live for that. The fee for the main event is $60 for non-members, $40 for members, $25 for students, free for children 10 and under. Science Sunday will be from 9 a.m. to 4 p.m. There will be workshops, and that'll cover topics such as microscopy, photography, and mushroom identification. Registration is also required for that, and it is $20 per person. Our speakers for the foray this year are Leon Chernoff and Michael Kuo. Leon is the editor of Mushroom the Journal, and his talk is going to be titled Life as a Mushroom, a Mushroom's Eye View of Life. And Michael's talk is going to be titled This Lecture Stinks, Fun with Phalloid Fungi. Sounds like a lot of fun. I'm really looking forward to these speakers. Also, a little note about getting to the Rose Barn. And someone who knows this better than me can feel free to jump in and explain, but basically this road here at Babcock Boulevard and Pierce Mill Road. So Pierce Mill Road is going to be one way only. So you'll only be able to get to the Rose Barn if you take this detour around and down. Did I explain that well enough?
Yeah, I think that was accurate. If you happen to be lucky enough to be coming from the north, you don't have to do that whole circle, but you can't go the usual way. We will be able to get into the Rose Barn parking lot and get back out. But if you're actually loading at the Rose Barn, then when you come out, you have to go that way, that one direction. So as I understand it, Fluff and Kara are trying to arrange all the walks such that you can go, back out on the 2-lane roads and stay away from the one-way street.
Correct. I'm trying. I
Yeah.
think I've got some good ones picked out. I think that we'll have it pretty well. I think they'll be convenient and good mushroom wants as well.
Good.
Yay. Awesome. And there will be more details on this detour in the newsletter that comes out in September. And there will be more information about it on our website as well. Speaking of the foray and other events, I wanted to put out a call for volunteers. So if anyone can volunteer at our next meeting, we could really use anyone who has extra shiitake log inoculation tools, if you have supplies for that, and can bring them about a half hour early, around 5:30, that would be awesome. Yeah. And also for the link-off foray, we've got openings for setup and cleanup, hospitality, cooks, walk leaders, and identifiers. And if you go to our website or newsletter, there is a link there to our Sign Up Genius where you can register for a volunteer slot. So we really appreciate everyone who volunteers for the club in different ways. And we wouldn't be able to do all of the awesome things we do if it wasn't for people's generosity. So thank you so much to everyone who has ever volunteered for the club. And if you're able to volunteer for either of these events, it is greatly appreciated. Here's a membership report. So we've gained 212 new members so far this year, which is just continuing to build a record-setting year for the club. So thank you so much to all of our members, new and old, for supporting the club. It is truly an honor to serve so many mushroom enthusiasts from Western Pennsylvania and beyond. So yay for us for being such a huge club of dedicated mushroom enthusiasts. And if you aren't already a member and you are watching this live on Facebook or recorded after the fact, here's some information on our dues and member benefits. Other benefits that aren't listed include receiving our bi-monthly newsletter, getting updates on walks and events, getting a discount on your registration for the link-off foray, and a mushroom grow kit if you're able to attend our meeting in August. Next up, I wanted to recognize some folks who submitted lists to earn a mushroom ID button. Our button ID program recognizes club members who can identify different species of fungi. Buttons are awarded at the 10, 25, 50, 75, 100, 150, and 300 level. Maybe even higher. Members can submit their list for verification to the email listed here. And if you're using iNaturalist or Mushroom Observer, that makes it a little easier to generate a list to submit it. Or you can just keep track of it yourself, however you see fit. But yeah, you can check out the button ID page on our website for more details. And tonight we have 2 members that we are recognizing for their mushroom identification efforts. So I wanted to recognize Cherie and Dan for submitting their lists. Yeah, so Cherie is going to be a new identifier for the club. So give a round of applause to these folks for submitting their lists. Yeah, it's awesome to see people continuing to learn how to identify mushrooms and build their skill. All right, and if you are looking to learn how to identify more fungi, and you have the time and want to stick around, after the speaker tonight, we will have our mushroom ID session. So Kara and Garrett will be on here helping folks identify any mushrooms that they can show us some pictures of. So if you have things posted to iNaturalist, that'll make it easier to view because you can just copy and paste the link into the chat, or share your screen pretty easily and pull up your photos. But yeah.
You can do a show and tell too if you just want to show some fun finds. Like, oh my God, look what I found. I'm happy to see your mushrooms.
I think I might have a couple show and tell things to share. So I'm excited.
Yay.
Yeah. So I already mentioned a little bit about next month's meeting. It's going to be our mushroom cultivation meeting with Rebecca Miller and friends. Yeah, meeting in person at Beechwood Farms. I'm just going to go ahead and say that masks are encouraged for the indoor portion of the meeting. Please be vaccinated if you're coming to our meetings. Just throwing that out there. We should all be getting vaccinated and taking care of ourselves and each other. But yeah, this year we will again have grow kits for oyster mushrooms, and we'll be inoculating shiitake logs. You must be a member to receive a kit. So bring your membership card and a clean plastic shopping bag to get your cottonseed hulls for your oyster mushrooms. Yeah, if you have access to a log for shiitake cultivation, you can bring one of those. Ideally, it is something that has been cut within the past 3 months. Older logs will already be colonized with wild fungi and will not produce shiitake mushrooms. Oak makes the best log, and maple will also work. And you want your log to be about 3 feet long and a diameter of 5 to 8 inches. And log drilling will begin at 6 p.m. and we'll take a break at 7 to go inside for announcements and cultivation instructions. In the event of heavy rain, thunder, or lightning, the log drilling will be rescheduled. So that's details on our next meeting. Whew, that was a lot. All right, any other questions or announcements before I introduce tonight's speakers? Just going back into the chat. Cool. I don't see any questions here. Well, without further ado, I will introduce Thomas Roehl, our speaker tonight, who will be giving a presentation on Flammulina velutipes. So Thomas is a scholarship grant recipient from our club, and he is pursuing a master's degree in biology at the University of Wisconsin-La Crosse, where he is advised by Todd Osmundson and Tom Volk. So yeah, Thomas, thanks so much for being here. I'll pass it over to you.
Okay, thank you for that introduction. So my presentation tonight is going to be about how mushrooms develop. And then specifically, I'm going to talk about my research into mushroom development in Flammulina velutipes. I have to, there we go. All right, so my presentation, I'm gonna first go into a little bit more about myself to introduce myself to you all, and then start with some background in developmental biology, then get into the big topic of mushroom development, and kind of slowly get into what my thesis research is about with Flammulina velutipes, and then specifically my thesis research. And actually near the end, I'm going to be talking about how mushroom cultivation works. So that will actually be a good preview of what you're going to get next month. Okay, so first about me. As was said, I am a master's student at the University of Wisconsin-La Crosse. I am nearing the end of my master's degree. All I have left to do is finish up my research and write my thesis. So I consider myself a mycologist, and specifically, I kind of go for cell biology as the methods I use. I'm also a mushroom hunter and a writer. So I have a blog, and I will actually provide a link to you for that at the end. I was born and raised in the Washington, D.C. area. So up here, and most of my mushroom knowledge comes from the Mid-Atlantic and Central Appalachian region of the country. However, I wasn't interested in mushrooms until I went to the University of Virginia for my undergraduate degree. And while there, I randomly took a class at Mountain Lake Biological Station about the biology of fungi. That actually started 10 years and 2 days ago. So this is an exciting anniversary for my love of mushrooms. After taking that class, my interest in the field really took off. And once I graduated, I joined the Mycological Association of Washington, where I was their newsletter editor for a few years. After that, I decided I wanted to go back to school and pursue a master's degree, which is how I ended up studying fungi at the University of Wisconsin-La Crosse. Okay, I think that's enough about me, so let's get started with some background on developmental biology. Developmental biology is the branch of science that studies how organisms go from simple cells to large, complicated structures. Most of the research in this field has been done on animals because animal systems can be directly applied to humans. So there's a lot of work that focuses on human developmental diseases and things like cancer and regenerative medicine. So there's a lot of push to focus on animals in this field, which means that we don't really get as much study about plant or fungal development. So if we don't have much research involving fungal development in this field, why would I bring it up? Well, it's actually, developmental biologists have identified several frameworks that are useful because they can be applied to all kinds of different organisms. Specifically, I would like to introduce you to 4 basic processes of mushroom development, or not mushroom development, development in general. So things an organism has to do in order to become a complex, large animal, or whatever it is. The first of these is pattern formation. So that's basically what it sounds like. Different areas of a simple organism has to figure out what they're going to be when they grow up. So in our organism here, we have a simple pattern established where this area at the top is going to be the head, and these 2 areas at the bottom are going to be the legs. Pattern formation typically happens very early on in development. For example, in fruit fly eggs, the basic pattern of front and back is actually established even before the egg is fertilized. So they happen very early on, and they decide a lot of what's going to go on in the organism as it develops. However, these are still just blobs of cells at this point. So we actually have to take those cells and make them into useful shapes. That is the process of morphogenesis, which literally means creating shape. So you see our head area is now shaped like a head, and we have a couple arms and legs. The other process that has to happen is growth. So organisms always start out small and they have to get bigger. This can get a little bit confusing if you talk about cell growth, because that does not fall under this process. We're interested in growth of the entire organism or a whole organ. So big picture growth, that's what's going on with this process. And our last process is cell differentiation. So how do cells become different from one another? And if you consider your brain cell versus, say, a fat cell, you can see why this is important. So your brain cell is long and skinny and has a bunch of fibers at either end, whereas fat cells are just large circular blobs. And brain cells and fat cells perform different functions, which is why they're shaped differently. So cell differentiation is important because it figures out what type of cell each cell is eventually going to turn into. I also have to talk a little bit about fungal cells and what their structure is, because they're very strange. So the basic unit of the fungal cell is the hypha. And hyphae are just cylindrical chains of cells, and they grow at the tip, and they can branch and fuse with one another to make networks of cells called the mycelium. So we have a picture of a mycelium on the right-hand side of the screen. And one thing you should note about it is it's a rather diffuse network. There's all kinds of gaps. It's not like all of the cells are squished together. So that was going to be important later on when we talk about mushrooms. So keep that in mind. The mycelium is rather diffuse network. One of the weird things about hyphae and the definition of a cell in fungi is that cells are broken up by these thin little walls called septa. And this is what a septum looks like. It's actually got a big hole in the middle. And because there's a big hole in the middle, that means anything in one cellular compartment can just move into the other cell. So the definition of cell in a fungus is really loose because all the cytoplasm just kind of flows wherever it wants to. That has some weird developmental consequences, most of which I'm not going to get into, but I would like to bring up a couple. So a couple weird things about fungal development. One is that development happens repeatedly throughout the life cycle of the fungus. So for example, I have some Amanita muscaria here, and these were all growing in a fairy ring. So they were all part of one single mycelium. And this mycelium is developing 4 separate times, and these are all in 4 different stages of development. And it can probably is going to produce mushrooms again next year. So we see development happening repeatedly throughout the life cycle. That's really strange because as animals, we only develop once. We start off as babies, we mature into adults, and that's it. We don't restart the process ever. So in this respect, fungi are more similar to plants in their development because plants also undergo repeated development. Every spring, they have to make new leaves, and every summer, they make new fruit. So there's developmental processes that recur throughout the life cycle of a plant. So from a developmental standpoint, fungi are more similar to plants, even though they're more closely related to animals. Another weird aspect of fungal development is that almost all fungal cells can revert back to an undifferentiated state. So what this means is that you can take a piece from a mushroom, put it on a petri dish, and instead of growing more mushroom cells, that piece of the mushroom is going to grow mycelium cells. This is really useful if you want to cultivate a mushroom, because all you have to do is take a piece of a mushroom and put it on a petri dish. But it's really strange from a developmental standpoint, because in plants and animals, cells don't readily do that. There was actually a Nobel Prize awarded for somebody who figured out how to do that with animal cells, but it's a very long and complicated and technically difficult process. Fungal cells are the bizarre ones because they just immediately can revert to an undifferentiated state and grow a new mycelium. Okay, that's enough of background on developmental biology and fungal cells. So now let's get started with the main idea for this presentation, mushroom development. Before I get into the specifics, I would like to spend some time asking the question, why should we study development in mushrooms? There's actually only 2 or 3 labs around the country that are studying mushroom development. So there's not a lot of research going into this, but I think there should be a lot more. First of all, it can tell us a lot about how mushrooms evolved. So the earliest fossilized mushroom we have is this one right here. Mushrooms typically don't fossilize very well because they're squishy, and squishy things don't tend to fossilize. So this one had to be preserved in amber to survive. And as you can see, it's already got a pileus and gills, and there's actually a little stipe on the underside of this mushroom. So it really looks like mushrooms we already see around us today. So if you want to ask, how did these structures come to be? The fossil record cannot answer that. So instead, we have to turn to other methods of research to figure out those answers. One of those is by studying development. So if we can figure out what genes are responsible for growing these different mushroom tissues, we can then start to look at how they differ among all branches of the fungal tree of life. And that will allow us to figure out how those specific genes evolved, which can then help us infer how the structures themselves evolved over time. Another good reason to study development is that it can teach us a lot about fungal cell biology. So fungal cell biology has mostly been studied in things like yeasts and mycelium. So we know a lot about how fungal cells interact with one another in this diffuse network of cells. But mushrooms are very different structures. In mushrooms, hyphae are closely stuffed together, and we don't really know how they communicate or coordinate their actions. And that coordination is very important to actually build a mushroom. I have an example in this picture here of what happens when mushroom coordination fails. So this mushroom was a super weird specimen where half of it was tall and then the pileus was split down the middle and the other half was kind of on this short branch. And it was just amazing. Somehow the coordination in this mushroom was interrupted and the whole mushroom just ripped itself in half. So what is driving that process that allowed it to lose communication? I have no idea because it hasn't really been studied yet. So there's a whole lot we can learn about how fungal cells behave specifically in mushrooms. Another important reason for studying mushroom development is, of course, the cultivation industry. Cultivation, the whole point is to have mushrooms successfully develop so you can sell them to people as food. And in fact, there have been a few instances where an interest in cultivation has driven research into development of mushrooms. One of the best examples is the mycovirus responsible for La France disease in Agaricus bisporus, where the mushrooms don't develop correctly, so of course then you can't sell them. So there's some interesting research involving that area. Other things you could do with mushrooms once you get a better understanding of how they work would be things like grow larger mushrooms or grow more precisely trigger the fruiting of mushrooms and so on. We're not nearly there yet, so this is all kind of far out in the future, if only we knew more ideas. Lastly, but certainly not least, mushroom hunters have always been interested in mushroom development. In addition to the fact that we're finding mushrooms, we also find weird stuff like my mushroom on the bottom here, which is supposed to be Udamanciella furfuracea, but if you know that mushroom, it looks nothing like that normally. So there's something weird going on with its development. Another example is rose combs on chanterelles. Why does the chanterelle decide to produce gill tissue on top of its cap? Nobody has a good answer for that because it really hasn't been studied. Even though we don't know much about mushroom development, we do know some, and what we do know can be summarized in this model of typical mushroom development. Mushroom development usually starts off when a ball of cells comes together, and that ball is called a fruit body initial. There doesn't have much structure to it. It's really just the basis around which the rest of the mushroom is going to be built. As development continues, hyphae grow into the fruit body initial and it becomes more dense and tissues start to appear. At that point, we call it a primordium. By the end of the primordial stage, all of the major tissues of the mushroom have already been established. You've probably seen this in Amanita buttons, where if you cut one in half, it's got a cap and stipe and gills already formed inside that circular structure. So that's the primordium. And because all of these tissues are already present, all that's left to do is expand that pattern. So this typically happens in the following order. The stipe usually elongates first, followed by the cap expanding, and lastly, the gills expand. So there's two major events happening in mushroom development. First is that pattern formation occurs in the primordium, and secondly, the patterns established are expanded to make the fully mature mushroom. And I'm going to go into a little bit more detail on each of those points. So let's start with pattern formation. As I mentioned, this happens in the primordium, and where it's been studied most rigorously is with forming gill tissue. So there's a really simple rule for early gill tissue formation. Where there is space, form a gill. Obviously, that rule is very loose, and it can result in a convoluted mess like you see here with from the early formation of Volvariella bombacina gills. So how do we go from this convoluted mess to an orderly linear gill tissue? That is actually the result of pileus expansion. So when you have the pileus expanding on the outside, it actually pulls and stretches the gill tissue. And that stretching action makes the gills line up in parallel ridges. You can actually see this happening when you fold laundry. So if you have a sheet and hold it between your hands and then pull, you see that linear ridges develop in that sheet. So the same physical process that results in those ridges on your sheets is what is responsible for stretching the gills into their nice linear patterns. Patterns haven't really been studied in the cap and gill tissue as much. So that's all I'm going to say about pattern formation. We still need more research in this area. Let's switch to expanding the mushroom. That whole process is driven specifically by cell expansion. And basically all of the cells get bigger, which causes the mushroom to inflate like a balloon. This actually satisfies two of our developmental processes, growth and morphogenesis. Let's start with growth because that's a little bit easier to understand. If you have a small mushroom and all of the cells inside it get bigger, the whole mushroom itself is going to get bigger. So that satisfies our process of growth. It's a little more difficult to see how that translates into morphogenesis because that is generating shape. But we have a good example of that with the mushroom Coprinopsis cinerea. So here on the bottom row of this diagram, we have on the left-hand side a primordial cross-section of the stipe, and on the right-hand side we have a fully mature cross-section of the stipe. And you can see the stipe of this mushroom has a hole in the middle of it, which is labeled lumen in this diagram. So how do we go from a solid stipe in the primordium to a stipe with a hole? That is also driven by cell expansion, but it's a specific subset of cells that are expanding. For Coprinopsis cinerea, we are going to expand this second to innermost layer of cells, which are gray in this diagram. As you can see in the primordium, this is a donut-shaped ring of cells. So if we have a donut shape and we make it bigger, it's still going to be a donut shape. So when looking at the right side of this diagram, those gray cells are specifically larger than all the other ones. They've expanded, but the other ones have not. And because the other ones in the center haven't expanded, they were kind of drawn out to the inner edge of the donut, which results, of course, in a gap in the middle of the mushroom. So we've satisfied morphogenesis because we have specifically created that shape of a hollow stipe just using the process of cell expansion. I've actually got a video of my Flammulina mushrooms expanding their cells. We have the cells in this video, you can tell that they're expanding because you probably can't see this over Zoom, but if you had the HD video, you would be able to look at the hairs along the stipe and see that even though the stipe is getting bigger, the hairs remain in relatively the same place. So the hairs near the top stay near the top, the hairs near the bottom stay near the bottom. So that shows you that the cells are expanding and there's not any new tissue being added. It looks like they're growing from the tip, but actually the cells themselves are getting bigger, which is making these things get taller. There's actually some really great videos of this process in the movie Fantastic Fungi, which somebody on Facebook said that that movie is coming to Netflix. So if you have a Netflix account and haven't seen that movie, now is a good time to go try and find it. And if you watch it, pay special attention to the time-lapse videos of Amanita muscaria growing, because you'll see that Amanita muscaria starts off small and has a bunch of dots on the top. And once it gets big, the dots have become equally spaced out on the cap. So the same thing would happen if you took a balloon, put some Sharpie points all over it, and then blew up the balloon. All of the marks that you made would get larger equally from one another. So that's a simple way you can tell just by looking at the arrangement of dots on top of the mushroom that its cells are expanding as the mushroom grows. All right, there is one major limitation with this model of development, and you might have guessed it based on the mushrooms I've been talking about. So I've talked about Coprinopsis cinerea, Volvariella bombacina, Flammulina velutipes, and Amanita muscaria. Those are all gilled mushrooms. And in fact, our model of development was created using research from only gilled mushrooms. That means our model might not apply to things like boletes or lion's mane or puffballs or polypores, because the data that went into the model didn't come from any of those other groups. And in fact, there's good reason to believe that this model of development doesn't apply in many cases. And that's because mycologists agree that there are two different types of mushroom development: determinate growth and indeterminate growth. Determinate growth can be recognized when mushrooms next to each other push aside other mushrooms and debris. So in our picture of agaricus here, you can see the mushroom on the right is pushing the mushroom on the left out of the way. So the stipe has ended up kind of off to the side of the pileus because the whole pileus was just kind of shoved to the side by our mushroom on the right. This type of development fits the model very well because if you imagine the mushroom as an inflating balloon, if you inflated the balloon nearby a bunch of other balloons, it would simply push the balloons out of the way. So determinate growth agrees with our model. Indeterminate growth, however, does something different. You can recognize this type of growth when mushrooms growing nearby each other meet and fuse with one another, and when they engulf debris into their fruit bodies. So you can't replicate that with a balloon. If you were to blow up a balloon next to it, another balloon, it simply can't fuse with the nearby balloon. So something different is happening in these mushrooms. I want to take a short look at this other process called haptomorphosis, which is just a fancy term that is used specifically when mushrooms grow around obstacles. And we can see this happening in two places on my Phalus schweinitzii picture. On the left here, a pine needle fell on top of our mushroom, and you can see right in the middle where the mushroom is starting to grow around, and eventually it will engulf the whole needle into its fruit body. And on the right-hand side, we have an example of where some grass blades were growing nearby, and the mushroom has simply grown around them and just kept on going. So both of these are examples of haptomorphosis, which is a process that occurs in indeterminate growth. Even though mycologists recognize only two types of growth, there are some weird examples of mushrooms that don't quite fit into either category. Take these boletes for an example. Our bolete on the right-hand side, if you look around the base, we can see some debris that was lifted up and kind of dropped to the side as these mushrooms grow. But if we look to the right, so the left-hand side, the mushrooms are kind of fused together at the top. So the right-hand side is behaving more like a determinate mushroom, whereas the left-hand mushrooms are behaving more like indeterminate mushrooms. What's going on here? I don't know, because boletes, as far as I can tell, have never been studied from a developmental point of view. So there's lots of examples that don't fit our standard mushroom development model. But this is what we have to work with right now. And fortunately for me, my mushroom research focuses on Flammulina velutipes, which is in fact a gilled mushroom. So this model does apply to my species. Speaking of Flammulina velutipes, I'd like to talk a little bit about the organism I'm using in my research. So you've probably seen this in the wild before, but if you don't know how to recognize it, here's what you should look for. It has a slimy orange pileus. The gills are pale yellow and they are attached to the stipe. They leave a white spore print. The stipe itself is probably the most beautiful part of this mushroom. It is a nice velvety black structure. Another thing to note is that the mushrooms form in dense clusters and that they grow on hardwood. Dead hardwood, I should specify. However, even though you find this nice umbrella-shaped mushroom in the wild, if you were to go to the grocery store and find these mushrooms, they look completely different. This form is, there it is. This form is called the enoki form. That's just the Japanese name for this mushroom. And it's kind of tall and spaghetti-like with a really tiny cap. And mushroom growers specifically alter the environment to produce this form. So how you grow it is that you fill some jars with a mixture of sawdust and grain, add some Flammulina mycelium, and then once it colonizes the jar, you put a collar around it. And that collar is important because it increases the concentration of carbon dioxide around the mushrooms, and it also reduces the amount of light available to the mushrooms. So here's my setup that I used in my research. I had to, there is a jar filled with a sawdust rye mixture here that you can't see because it's covered in aluminum foil. And then to do the collar, I just used an old tin can because it fit perfectly on the side or on top of my jar. And I had to add an extra jar on top to reduce light even further because my light was coming in from above, so that one can used as a collar didn't quite do the trick. So why do we care about raising carbon dioxide and lowering light? Well, those are actually two environmental cues that can be found in nature that cause this spaghetti-like version of the mushroom to be produced. And I have a beautiful picture of this from the Western Pennsylvania Club's own Cecily Franklin. She has this stump on her property and found some Flammulina growing over it, and then removed the bark that was blocking part of the mushrooms to reveal our cultivated form growing right there in the wild. So the cultivated form is actually a response to conditions found underneath bark. Obviously, the point of a mushroom is to release spores into the air. The mushroom can't do that if it's trapped underneath bark. But sometimes the fruit body initials form underneath bark. So Flammulina has developed this spaghetti-like growth form as a way to escape the confines of bark. So if it finds itself in a bark-like environment where it's dark and there's a lot of carbon dioxide, it will grow in this really long form. All of its resource is devoted to growing upward. It doesn't bother with developing any color or that beautiful fuzzy stipe. It just wants to go up. And once it goes up, it hopefully will find a crack in the bark where it can sneak out and access the external air. And at that point, it switches to growing in our normal umbrella-shaped form. So that specific difference between our two growth forms is really the reason I decided to study this mushroom. Specifically from a developmental point of view, if you can find a difference in a single mushroom that is caused by a specific set of known conditions, that makes for a useful comparison later on. Another good reason for studying this mushroom is that it's easy to grow and it has a sequenced genome. Because it has a sequenced genome, I can actually skip one of the steps later on in my analysis, and that will save me some time on my research. And lastly, it's also a commercially relevant mushroom. So hopefully one day what I'm studying will lead to some benefits for the mushroom cultivation industry. So my research specifically is looking at cell differentiation in Flammulina velutipes. And just as a reminder, cell differentiation is our fourth basic developmental process. I haven't talked about it yet, so it is how cells become different from one another. And we can see just by looking at the Flammulina mushrooms that the cells are different from one another. the cap is orange, but the stipe is black, for example. So their cells are specifically different. I want to know what is causing those differences. And to do that, I need to look at what's going on inside the cells. Before I get into too much detail about that, I have to explain a bit of what this concept of gene expression is. If you remember back to some high school biology, you can probably know that all of the information for your cells is contained on molecules of DNA that are found inside the cell's nucleus. So DNA, unfortunately, cannot go out into the cytoplasm, which is where cell activity occurs. So we have to have this secondary molecule that takes the information stored in your genes from DNA out into the rest of the cell where it can actually be put to use. That molecule is called mRNA. So the first thing that happens is that mRNA copies down instructions found on DNA. It then moves out of the nucleus into the cytoplasm of the cell, where its instructions are read by a structure called a ribosome. As it reads the information, it forms this chain of amino acids. And that chain is then broken off and folded into a protein. And proteins are the they're the machinery that allows the rest of the cell to run. So ultimately, all the instructions on DNA have to be converted into proteins for the cell to work. So we can figure out what a cell does by studying all of these 3 steps in our protein production process. If we study DNA, that's called genomics. And unfortunately, it's not very useful for cell differentiation because all of the cells have the same genome. So instead, we have to study our later processes in this pathway. If we look at mRNA, that is called transcriptomics, and if we consider proteins, that is proteomics. Both transcriptomics and proteomics can tell us which genes are active at any given time. This is important because obviously your cells don't use all of the genes in your genome. If you think about your brain cells, again, they don't want to be producing genes for fat cells, because if you had a bunch of fat building up in your brain, that would cause problems and make your brain stop working. So your brain cells specifically express genes that it needs to make proteins involved in brain function, whereas your fat cells specifically produce genes and proteins that are involved in the production of fat and utilization of energy. So different cells doing different things is the whole basis of a cell differentiation. And in order to figure out what's going on, we have to use either mRNA or proteins. Of course, I'm not the first person to look at gene expression in Flammulina velutipes. That honor goes to Sakamoto et al., who in 2002 ground up some mushrooms and mycelium and then spread the proteins in each across a gel. So here is what they got. Each of these dark blobs is a different protein. And the larger the blob, the more protein there is. So they compared these gels from the mushrooms to the mycelium and found 22 blobs that were different sizes. So they discovered that, yes, in fact, as the mushrooms grow, they are changing what proteins they're expressing. So there is gene expression, and it is correlated to development and cells becoming different from one another. Of course, in 2002, protein sequencing technology was virtually non-existent. So although they found 22 different differences, they had no idea what any of those differences were. If we fast forward to 2014, this study instead used mRNA, and they were able to sequence every single gene in the genome and figure out what gene was changing. So if, for example, we look at this hydrophobin FVH1, this whole graph here is called a heat map. And basically, the red areas show high gene expression, and the green areas show low gene expression. So where it's red, there was a lot of mRNA in that cell. And where it was green, there was no or almost no mRNA for that specific gene. So hydrophobin FVH1 is this specific row, and we can see there's a lot of it expressed in the mycelium, but it's not very present in the fruit body or the primordium. So now we can tell there is a difference, and we can quantify what that difference is and assign a specific gene causing that difference. In 3 years after that, we had a study that went back to proteins. They could sequence proteins now, so they were able to, again, identify which genes were expressed at different levels, but they went a step further and then figured out what each of those genes did. So in the pie chart on the left, this is all of the cellular processes that changed when a mushroom that were expressed more highly in the mushrooms. So you can see things like ribosomes and nucleus maintenance and proteasome complex, that's basically a recycling center for the cell. All of these are more active in the mushrooms. And on the right-hand side, we have the same pie chart, but for cell processes that are less active in the mushrooms. So these types of analyses are then getting at what the cell is doing differently, not just what genes are different. All right, so here is the whole list of all of the studies that have looked at enoki gene expression. If we look on the right-hand side of this column, we see that, not surprisingly, they started out looking at very small numbers of genes and then got more and more and more as time went on. Flammulina has about 12,000 genes in its genome, so this study with mRNA was able to sequence nearly every single gene. Our protein study, since it's still difficult to sequence proteins, only sampled about 10% of the genes in Flammulina velutipes. So if you want to know everything that's going on, you have to use mRNA to study that. Another thing you should note for this table is that basically everybody has studied the mycelium, and that's because the mycelium is obviously not a mushroom. So genes that are active in the mycelium are probably not important for mushroom development. So the mycelium is a nice baseline to compare everything to. The primordium has also been studied by most researchers, but surprisingly, nobody has looked at the tissues in the primordium. As I mentioned earlier, we already have the cap and stipe and gills formed inside the primordium. So if you just look at it as one structure, you're missing all of the gene expression differences that could exist between those different tissues. There are 2 studies that did consider tissues, but neither study has looked at gill tissue, which is kind of unusual because that's probably the most important part of the mushroom. It does produce the spores after all. Looking at the top of this table, you can see that the mycelium, primordium, young mushrooms, and cultivated mushrooms have been studied, but nobody's ever studied the fully grown umbrella-shaped mushrooms, which I found surprising. That's probably the first stage that I would have tried to study. So I hope to fill in some of these gaps with my research that have not been studied. So for my thesis project, what exactly am I doing? I'm going to try and sequence all of the tissues in Flammulina velutipes and figure out what genes are expressed in all different stages. I have 3 main objectives: to identify gene expression differences between different tissues, within each tissue as development progresses, and between the normal and cultivated forms. So by filling in those objectives, I should give a broader picture of what's happening throughout mushroom development in this species. The first thing I had to do for this project was to collect a sample from the wild. This is an actual picture of the mushroom I used. Isn't it beautiful? And what I did was I took it back to the lab and then took a piece off of it and cultured it on a petri dish. So for a while, I just maintained the mycelium growing on different petri dishes. Once I had some fully colonized petri dishes, I could go on to the next step, which is the spawn run. To start the spawn run, you first have to have a jar of sterilized rye grain, and then you cut up a petri dish and put the pieces into that jar. You have to shake the jar every few days to make sure that the rye grains don't stick together. At the end of this stage, you should end up with a jar that looks like the picture on the right, where everything, all of the rye grains are coated in a nice white fuzz. That demonstrates that the mycelium for Flammulina has been dispersed evenly throughout the jar. So incidentally, I did all of my sterilization in my apartment using this pressure cooker that one of my fellow students loaned me. I started out using the university's autoclave, but at one point it did not successfully sterilize any of my jars. And that set me back about 2 months where I had to go back to earlier material and try and get it back into culture. So that was a big mess. And I decided to ignore the fancy research equipment in the lab and instead use stuff that works out of my home. And actually with the pressure cooker, I have a 100% success rate for sterilizing my jars. Okay, once you have a fully colonized spawn jar, you can go to the next step, which is transferring it to the fruiting mixture. So the fruiting mixture is a mixture of sawdust and rye grain. Flammulina grows on hardwood, so you need a hardwood sawdust, and that provides most of the carbon for the mushroom. But you have to add in a little extra rye grain as a nitrogen source, which really helps the fungus produce mushrooms specifically. So what you do, you sterilize your sawdust rye mixture, and then you add some of the colonized spawn to that jar and then shake it up. And after a few days, you should get something like this, where we have a polka dot pattern of these white splotches. So each of these white splotches is where one of our colonized rye grains landed, and then mycelium has started growing out of that rye grain. We want a nice polka dot pattern because that will allow the fungus to quickly and easily colonize the whole jar. And once your jar is completely colonized, you can go on to the next step, which is actually fruiting the mushrooms. So here is the fruiting chamber that I built for myself in my apartment again. And this was my pandemic arts and crafts activity. So at the time, the university was completely shut down, and I had to come up with another way to make sure I could continue doing my research, even without access to the lab. And this is what I came up with. It looks complicated, but basically it is just an ice water bath, and the jars of mushrooms float in the middle of that. It's important to have an ice water bath because for Flammulina specifically, it doesn't produce mushrooms if the temperature is above 60 degrees. Another important feature is the overhead lamp. That was the only direction that I could get light into my water bath. And having a light source is important because that stimulates fruit body initial formation. So we need a 60 degrees or lower temperature and a light source in order for our Flammulina to make mushrooms. Once the mushrooms started forming, I could then add a jar or add a collar to one of my jars to make sure that it formed the cultivated version of the mushrooms. And I showed you a picture of that earlier in my presentation. Once I had mushrooms growing and they reached the appropriate stages, I could begin the sampling process. So for my sampling, I was interested in 4 different stages: the primordium, the mature cultivated form mushrooms, the young normal mushrooms, and the mature normal mushrooms. In each of these different stages, I sampled 4 tissues: the mycelium, the stipe, the gills, and the pileus. I repeated this process 3 separate times, so I had a total, of 48 samples, 4 different stages times 4 types of tissue times 3 replicates. So here's what that actually looks like in real life. This is a picture of the fruit body initials from Flammulina. You can see that they're balls of cells and they form around this amber colored droplet of liquid. Now, I didn't sample these because they were way too small for the tools that I had available. The next stage is the primordium. And in Flammulina, the primordia are kind of triangular in shape. And at the very tip of the triangle, you have the tiny cap and gills. So sampling this, I wish I'd gotten a picture of this, but I could not physically do this. I had a razor blade in each hand and was looking down through a dissecting scope and carefully cutting off tiny slices of a mushroom. So taking a picture was out of the question. The next stage I sampled was the young mushrooms, which looked very similar to the primordia, except that the stipe has begun elongating and the edge of the cap has detached from the stipe. This was also very difficult to sample because they're still really small at this point. The next stage is the cultivated form, which again, it still looks pretty similar to our young mushrooms. It's just got a very, very long stipe and a slightly bigger pileus. Our final stage that I sampled was the mature normal version of the mushroom, which you can see the gills have developed and it's got a large pileus. And on the right-hand side, we see a couple that have even gotten the dark stipe going on. So those are the ones that I actually took samples of. This was probably the easiest stage to sample because it was just so much bigger and easier to work with. Once I took a sample from each of the tissues, I had to immediately put it in a tiny tube and freeze it in liquid nitrogen. And that was to ensure that the mRNA was preserved. Next, after I had all of my samples collected, I could start the process called RNA-seq. And this is just a method that allows you to sequence all of the mRNA in a cell. So the first step of this process is to get your frozen samples and pulverize them. So I had to crush the samples to release the contents of their cells. Next, I had to extract the RNA from that sample, and I just used a kit to do that, which was fairly simple. And that's as far as I've gotten in my research so far. I'm still waiting on some kits to arrive for the next part of the process. So stay tuned for results later on. The next thing after you extract the RNA is to purify the mRNA. And that gets rid of a type of RNA we don't want, which is called rRNA. Once we have the purified mRNA, we can go to the next step, which is creating a copy of that on DNA. So this cDNA, as it's called, the C just stands for copy, is just a copy of the mRNA that we have. And we do that because there's already so many tools that exist for working with DNA. So it's just a lot easier to convert everything into DNA. Once we have our DNA copies, we have to add some pieces to the ends that are called barcoded Illumina adapters. The barcoded part of this just means that I can figure out which sample each piece of DNA came from. The Illumina adapters part is just a specific tool that allows it to interface with this DNA sequencing technology. Once we have that assembled, we can go to the next step, which is a couple rounds of PCR, and that just creates more copies of our cDNA. That's important to make sure we have enough material to submit to the sequencing lab. Afterward, the final step is submitting it for sequencing, and I will be sequencing using Illumina, and that is going to return an enormous data file with a whole bunch of A's, T's, G's, and C's in it. Now, that data cannot be read by a normal human, so we need to turn to some computer programs in order to figure out what that means. Specifically, I'm going to be using 3 programs in the new tuxedo pathway: HiSat, StringTie, and BallGown. Our first one, HiSat, just matches each sequence with its corresponding gene. StringTie figures out, uses those matches to estimate how much mRNA was present in the original sample. And BallGown then uses the information from StringTie and calculates gene expression levels. Once we've run BallGown, we can use the data to generate a heat map like this. And that will allow us to pick out interesting genes that are doing something different, like our old friend hydrophobin FVH1. After we have identified these genes, we can run them through 2 different databases, which can give us more information about what that gene does. The gene ontology database will tell us the functions of each gene. So in this case, hydrophobins are little proteins that coat the surface of the fungal cell wall. So it would have a function in the extracellular region. If we look it up in the database called the Kyoto Encyclopedia of Genes and Genomes, that tells us what pathways our gene is involved with. In this case, since it's kind of a structural component of the cell wall, it's not really involved in anything like cell metabolism, energy utilization, protein formation. It doesn't do any of that. It's just a structural component. So it would give us back something like none. Both of these two pieces of data tell us more about what the cell is actually doing. So once we have all of this data together, I hope to use it to come up with 4 different outcomes. First, I will end up with detailed tissue level gene expression. Specifically, I will have gene expression in the gill tissue and the mature normal mushrooms, which nobody's looked at before. So I will have some interesting contribution to the field. Don't know what it will be, but it will be interesting. Also, we can compare all the different tissues and mushroom stages with one another to identify gene expression patterns associated with each specific tissue, and hopefully find some key development-related genes. All right, so I've done sampling, I've started RNA-seq, and that's as far as I've gotten, but I still have some other interesting observations from my project so far. First of all, I mentioned earlier, Flammulina forms fruit body initials around these amber-colored droplets, and that turns out to be very useful if you want to study fruit body initials. Most of the mushrooms used for development research form white fruit body initials on top of a white mycelium. So it's nearly impossible to find them because they're tiny white balls of cells growing on top of a fuzzy lawn of cells. It's just not easy. Flammulina, however, because of that dark color that they form around, you can pick them out immediately just using your naked eye. So for people who are interested in studying these really early stages of mushroom formation, Flammulina would be a great organism to do more of that work in. Another interesting thing I found is that sometimes new mushrooms will form on top of an old stipe. I have no idea what's going on there, but from a developmental perspective, that is very weird. It would be like you forming a whole nother body off of your arm. That just doesn't happen. So I don't know what's going on, but I would love to study that phenomenon because it is very strange from a developmental standpoint. The last interesting thing I found was, especially later on in my mushroom growing, I found some of the mushrooms had this development altering disease. So if you look at the picture on the bottom right, the right-hand mushroom is a normal mushroom. It's got normal knife edge gills on it. But the mushroom on the left has these odd, blunted and thickened gills. I'm not sure what is causing this disease. I kind of suspect it is a virus, but I haven't quite ruled out some other things yet. So I'm hopefully we'll be able to figure out what's going on with this, because again, anytime you have a development altering disease, it provides a useful point of comparison to help figure out what goes on in the normal mushrooms. So all of these kind of observations just add to the value of Flammulina as a model system for studying mushroom development. All right, I know I provided you with a lot of information, so I would like to kind of review the key things I would like you to keep in mind after this presentation. First of all, we have some really cool research tools that you can use to figure out what each cell in a mushroom is doing. And this allows you to do some interesting comparisons, like comparing the spaghetti form of enoki to the normal umbrella-shaped form that's found in the wild. Another thing you should remember is our basic model of mushroom development, where the first thing that happens is a fruit body initial forms, then pattern formation occurs in the primordium, and lastly, the whole mushroom then expands as like a balloon. Of course, this model does not apply in all cases because we have two different types of growth: determinate growth, where the mushrooms push each other aside and push debris out of the way, and indeterminate growth, where mushrooms fuse with one another and grow around debris. Another thing I would like you to take away from this presentation is that you are mycologists. Obviously, I did a lot of my work out of my apartment using just random stuff I had lying around and not very complicated lab tools. So it is one of the coolest things about mycology that amateurs can make really big contributions to our knowledge of fungi. So I hope you all remember that you don't have to have a fancy degree or fancy lab equipment to make a contribution to mycology. Just keep your eyes open when you're going out on forays and hunting mushrooms. You never know what you're going to come across. And there is so much left to learn in the field of mycology. Development has mostly focused on guild mushrooms, which means that there's so many other morphological forms for us to consider. And even just basic fungal biology, you can learn things by walking through the forest that we don't just see in inside labs. So I hope you all take that to heart and make useful contributions to the field of science as you're, finding mushrooms to eat. All right, that is all I have for you. I have a couple slides of the resources that I use to reference in my presentation, but I know that's not very useful for you all. So I have a couple other resources you can use if you want to learn more about mushroom development. First of all, I have my blog, Fungus Fact Friday, and if you copy down the link on this screen or scan the QR code, it will take you to a page where I have collected all of my posts about mushroom development. Right now, there's only one in there, but I hope to add at least 3 more in the coming weeks. Another good resource is Fungal Morphogenesis by David Moore. That book was published in 1998 and is still the most complete resource we have on mushroom development. So if you really want to get into the weeds, see if you can find a copy of this book. Hopefully somebody will come along and update this with a new book, but that has not happened yet. I have to acknowledge a lot of the support that I've gotten in order to conduct my research. Specifically, I want to acknowledge my advisors, Dr. Todd Osmondson and Dr. Tom Volk. I also have to thank Dylan for the use of his pressure cooker and Beth for providing me with extra Petri dishes. I borrowed a lot of those from her. I also would like to thank everybody who got me liquid nitrogen. So far, that has been the most difficult process of my research. The system at UWL is just so weirdly complicated. It was a pain to navigate that. Of course, I also have to acknowledge everybody who's given me financial support for this project: the University of Wisconsin-La Crosse, the Sonoma County Mycological Association, the Mycological Association of Washington, D.C., and all of you at the Western Pennsylvania Mushroom Club. I haven't actually spent any of the money you've given me yet. It is all going to go toward the sequencing step. Sequencing, it turns out, is very expensive, and I wouldn't be able to do as much sequencing as I will be without your generous support. So thank you all very much for supporting this work. That is all I have. Do you all have any questions? I think some have come in through the chat. So let me see if I can get that up.
Yeah, thank you so much for this presentation, Thomas. That was wonderful. So yeah, there was a question up in the chat.
First one was Julie's.
Yeah, so you were talking about some of the research on cell development, and the question was like, is the idea of cells reverting being studied for cancer research? I don't know if you know who is studying that.
There was, I guess it's a slightly different process, but it's all kind of connected from the developmental biology standpoint, where normally cells start out as embryonic stem cells, which can form all different types of cells. But then as they go on, they specifically end up being a specific type of cell. For example, your skin cell. And your skin cells can only make more skin cells unless they become cancer cells. So in that case, some of the controls that prevent cells from doing things they're not supposed to get turned off and they go haywire. So developmental biology, a lot of the effort has been on figuring out what controls prevent cells from doing what they're not supposed to. And that kind of gets into the reversions with cancer work. Again, most of that's done in animals. It hasn't really been studied in fungi, but I wish somebody would study it because it's really fascinating.
Awesome. And then one more question in the chat, which is, do you know of any undergrad mycological research opportunities students can get involved in?
Offhand, no. And that would depend on your specific program. But I guess my best advice is to go on your university's website and look at the research areas that all the professors at the university are researching, and see if you can find something that is kind of interesting to you. I know that was one thing that I struggled with personally as an undergraduate. It didn't help that only one person at the university was kind of studying fungi. But there is definitely opportunities for undergraduates to get involved. So for example, at UWL, one of my colleagues, Dylan, he needed some extra funding for his project. So he had to bring on some undergrads, and the university funded the undergrads, which then went into helping his sequencing project. And they also had like a specific version of his project that they worked on. So there's definitely situations like that that exist, and people would be glad to have undergraduates help them. You just kind of have to suck it up and go talk to people, which I know is the hardest part for me. Yeah, if I could have, I wish that I could have created a better resource for connecting people to opportunities like that, but I didn't have time, and the pandemic kind of took over things.
Hey, Thomas, Richard Jacob had a comment. I don't know if you know this, but Richard has a PhD in proteomics. Yeah, so he's old school, I guess.
Yeah, well,
there he is.
There's probably a lot that I kind of glossed over that he would nitpick with, but I didn't want to get bogged down in the details. So there's a lot of nuance to what I said. I'm sure he knows better than I do how some of this works, actually.
It was just fun to see the 2D jail slide.
Is that the thing with the little blobs on the little black dots?
Yeah, so my PhD was automating the analysis of 2D jails. So using robotics and stuff. Yeah, back in the day.
Yeah, it's progressed a little bit since then, and we can now actually sequence proteins to some extent. I'm a little bit hazy on how that whole process works, but still going on. I think there's some really interesting stuff in that field that's going to come out in the next few years.
The big key is the cost of sequencing has gone down for whole genomes.
Yeah.
And now that the whole genomes are available, then you can actually identify the proteins a lot easier. Before, you get the protein sequence or amino acid sequence, but you wouldn't know what it was from. So a 2D gel for an unsequenced mushroom, 20 years ago, that would have been really hard work to get anywhere. But yeah, we're looking forward to the results of the mRNA sequencing. That will be, we're
yeah,
really
I know. I was hoping to have them done by this point, but various things got in the way that kept pushing it back. Like, oh no, you can't buy stuff for these 2 weeks because it's too close to the end of the fiscal year, and random stuff like that.
Yeah, I know how academia goes.
Well, wonderful. It doesn't look like we have any other questions, but I'll pause for one more moment just in case someone does. But yeah, thank you so much, Thomas, for your presentation. And really looking forward to the results from your research.
Thanks, I am too.
All right, so I think at this point, I will stop our live stream.